A very good morning to one and all. On behalf of management, principal, staff of Vignan Pharmacy College, cordially inviting resource person, principals, professors, and faculty members from various universities and institutions, 1 week online faculty development program on emerging trends in drug design and development. The main objectives of the FDP are participants can acquire basic knowledge on the latest trends of drug discovery and development to inculcate and enhance the abilities of the faculty and research scholars in the fostering knowledge, the domain of pharmaceutical chemistry research, able to utilize applications of artificial intelligence in research and development. In this faculty development program, talks are delivered by eminent scientists and professors from international central universities and national institutions. So I request all the participants to attend all the sessions without fail and get benefited. Thank you.
Namaste ji.
How are you, sir? So, okay, sir. I'm very happy, and first I would like to congratulate Rajalakshmi madam for making this EFDP program for all the faculty members. So, Sir Bharatam sir have a vast experience on the medicinal chemistry. So, I'm very happy. On behalf of Vignan Pharmacy College, we invite you, sir, for this EFDP seminar. So I request the coordinator, Dr. Rajalakshmi madam, to continue the further proceedings. Thank you.
Namaste, sir. Prasad ji.
Namaste ji.
Sir, now you can start the session, sir. So in spite of your busy schedule, you have accepted our invitation. Thank you very much, sir. Now you can proceed, sir.
Now I am going to go to a specific topic called structure-based drug design, PPR gamma agonist design. These are two different titles I have combined. Structure-based drug design is a topic of pharmacoinformatics. PPR gamma agonist design is designing a particular agonist against a particular target. So I have combined one applied title and one scientific title in this so that it is of use to every student. Okay, this is my name. All of you probably are aware. Here your Dr. Ramalakshmi introduced me to all of you. This is my address and you are welcome to Naipar Mohali to learn this. You can send your students to summer school. You are also welcome to participate in summer school. In summer school, during June and July, we give 50 days of training on this topic. You are welcome. Every year we give this training. Advertisement for this comes in April. So I am inviting all of you to visit Naipar website and apply and encourage your students to apply for this. Okay. Now, before we go into this, before I really go to the topic, I will touch upon the PPR gamma. What is PPR gamma? It is a target. It is a macromolecule. We want to design an agonist for that. What is an agonist? Agonist is a compound which activates the particular protein and it introduces some kind of conformational change. Conformational change is one of the very important characteristics that is happening. That is what is going to tell whether a particular organism is alive or not. For example, if you want to know whether I am alive or not, if I am changing my hands, if I am talking to you, if I am showing some kind of dynamism, you will think that I am alive. If I am looking like I am not moving at all, okay, you may think that this is not alive. Okay, exactly. Similarly, alive versus So for that, dynamism is important. Dynamism comes from the conformational changes. Conformational changes are always happening inside the body. Every molecule, every drug, whether it is inside the tablet or whether it is inside the stomach, whether it is inside the liver, whether it is inside the kidney, or it has reached the brain, wherever the drug is present, it is dynamically present. Dynamical in the sense it is always undergoing some kind of conformational change. This conformational change that is always happening. PPR gamma is undergoing conformational change, and PPR gamma agonist is undergoing conformational change. And because of this conformational change, signal is being transduced, and this signal is being generated and transduced across the membranes and across the body fluids. And these signals sometimes molecules, sometimes simple electronic signals. These signals are responsible for the therapeutic action. So PPR gamma is a macromolecule which is showing agonistic activity. That means it is showing the specific conformational change. We want to find we want to find a agonist. That means we want to activate the conformational changes happening in PPR gamma. That is the first step. Before going there, let us try to understand what is diabetes mellitus. If I start giving lecture on this, you will say we already know about it. Why should I give lecture on diabetes? So I will not say anything about it. We all know about it. It is the only non-infectious disease designated as an epidemic by World Health Organization. This is very prevalent, especially in our Andhra Pradesh. It is very, very prevalent. Most of us are using, every household is using metformin for sure. Okay. For diabetes mellitus, I will not tell much. I will go to PPR gamma. PPR gamma is a protein. It has got a 3D structure. This 3D structure is very important. We should always think in 3D. If I want to look at what is this object, you know very easily you can mention what is this. Because I can show it in 3D, I can talk. If I start describing it, there is an object with me. It has got power to punch. It has got two rods through which there are pins, and there is a. If I start describing a paragraph, you will not understand easily. If I show it in 3D, we can understand very easily. We know from our childhood, a picture is worth 1,000 words. But a picture in 3D is worth billion words. There is nothing like that. So if I can think about PPR gamma in 3 dimensions, if I can think about the drug molecule called rosiglitazone, which is interacting with PPR gamma in 3 dimensions, that means I am having a knowledge which is at least a billion times more than reading a paragraph on that. So that is why 3D thinking is important. So my lecture will always invite you to 3D thinking. Many times I am going to invite you during this lecture. So first thing I am going to do is think about PPR gamma in 3 dimensions. Okay, this is one thing. PPR alpha, PPR gamma, PPR delta. All the three are proteins. They are very similar. What are these? Peroxisome proliferating activator receptor. This is a activator receptor. It is receiving an activator. What is it doing? Proliferating peroxisome. This is a very biologically involved definition, but simpler definition we can take. PPAR. There are three varieties: PPR alpha, gamma, and delta. Then you may ask why beta is not there. Beta and delta are the same. Initially, they thought 4, but they converted it. They realized that beta is nothing but delta. So delta, we have so much knowledge. So let us forget about beta. This is how science evolves. Anyway, this is about the macromolecule. Let us talk about the small molecules. Small molecules which interact with PPR gamma are thiazolidine dione derivatives, oxazolidine dione derivatives, tyrosine derivatives, alkoxyphenyl propanoic acid derivatives, oxybutyric acid derivatives. There are so many derivatives, so many small molecules which can interact with the PPR gamma. Very similar. Let us take this as my hand as a macromolecule. I can introduce a small molecule. I can introduce. This is where a small molecule interacts with the macromolecule. That is exactly what happens in reality if we think in 3 dimensions. Okay, bigonides. We know some of those anti-diabetic molecules. Bigonides, metformin is a very important one. Beta adrenergic receptors, PTP1B inhibitors, GSK3 inhibitors. In all cases, there is a macromolecule which is having a 3D structure. There are many small molecules which is having 3D, which are having 3D structures. So if we can think on 3 dimensions, we can easily do drug discovery. For thinking in 3 dimensions, molecular modeling is helping. The computer graphics is helping. Your laptop is helping. Your desktop is helping. So if that these particular facilities are allowing us to think in 3D. 3D. We have got to think in 3D. Okay, suppose some of you probably have visited Naipur Mohali. Suppose I send you a photograph of Naipur Mohali. You will get some idea. Suppose I send the 3D, I will send a video of Naipur Mohali. You will understand better. I will send a 3D video of Naipur Mohali. You will understand in a much better way compared to that. Exactly the same way, I need to think about the small molecules in 3 dimensions. I need to think about the macromolecules in 3 dimensions. For that, computer-aided drug design is the best source. It is the only source. There may be experimentally something is possible, like protein crystallography, small molecule crystallography. These techniques are there, but on how many molecules you can do that? Maybe two, three, maybe 20. If you are a good scientist, if you are a great scientist, you may have access to 1 lakh macromolecules in protein data bank and maybe uh, billions of molecules. But can you have the 3D structure of the interaction between these two? Very easily. And how many cases you can create that? In how many cases you will have atomic level information, surface level information, electronic level information, and all others. Okay, let us think about it. Let us think about one of our friend who is attending this conference. One of our professor. Let me just take, for example, I am taking the name of one professor just for fun. Professor Anand. He is attending this symposium. Professor Anand, do you know him? But if you meet him, if you see him in 3 dimensions, if you shake hand with him, if you know how beautifully he smiles, how happily he can teach, how enthusiastically he interacts with his students, how beautifully he encourages his students, you will, if you observe that in 3 dimensions, you will realize, oh, okay, I have to learn some things from Professor Anand. Okay, there are some values available with him. I need to inculcate the same values when I am interacting with my student. But if I send you the photograph of the student, of Professor Anand, you may not be able to understand all of them. Exactly the same thing I am telling about my PPR gamma, about my PFDHFR, or about my bedaquiline, about my topo 2 isomerase, about any macromolecule, about every cell, about every antibody, about every virus. For example, SARS-CoV-2. Do I have three-dimensional information? If I can develop that, I can do a lot of research. That is where computers are directly and immediately available to us. Okay, so what I am trying to invite you is to think in three dimensions. Use your computers. You can use our computer very easily. We are buying computers. Maybe we are investing 40,000 rupees or 80,000 rupees. Your cousin or your son or your brother is demanding, please buy a computer laptop for me. It is very important for my classroom. But what he is doing, you can see very well. He may be watching movies, he may be watching videos, and he is using very efficiently computer only. He is sitting all the time in front of computer. Is he using computer for doing drug discovery? If he is not doing, take that computer and do it yourself and teach your students in the classroom. Computer-aided drug design can be done very easily with the facilities that you already have. There are some software available free of cost. PyMol is for macromolecular modeling. Okay, you download it; it is free. There are several other small molecular modeling software. You download them, very easy. Free, simply type computer-aided drug design in molecular modeling free software. That's all. You will get a list. Whatever. You download each one of them. Start learning slowly. Think in three dimensions. This is very, very important for any successful student. Teach this message to all the students in your class, every batch of students, that thinking in three dimensions is important. So that's what I'm going to teach you in this lecture. How to think in three dimensions. How did we do it in Naipur Mohali? This is what we have looked at, first thing. The very first thing we look at it is how the molecule looks like. This is a 2D structure of the molecule. If we divide this molecule, which is actually called nothing but rosiglitazone, it is a drug molecule which is binding to PPR gamma. Now, my message is that this molecule has got so many bonds. It has got so many rings. There is a ring called phenyl ring in the middle. There is another ring on the left hand, right hand side called the thiazolidine dione ring, sulfur nitrogen containing ring, which has got two carbonyl groups. This is a ring. And then on the left hand side, you have a pyridine ring. And then in between, there are several rotatable single bonds. Carbon, nitrogen, carbon, nitrogen, carbon, 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 oxygen, rotatable bonds. These are called linkers. And then there are pharmacophoric features. The pharmacophoric features are not just drugs, now are not just structural features. Phenyl group is not a pharmacophoric feature. Phenyl group is a structure related phenomenon. Phenyl group is a hydrophobic center. Thiazolidine dione is an acidic center. That is a pharmacophoric feature. Pyridine ring is a lipophilic center. That is a pharmacophoric feature. So looking at the 2D structure of my molecule, I can find out which are the pharmacophoric features. This is nothing very strange. This is what we do. This is a important requirement, and we do it every day. We do very well in our classroom. When our students enter the classroom, we know who is mischievous and who is silent and who is attentive in our class. When we are making an effort for 1 hour, who has understood our lecture, who has not understood our lecture, we can easily guess. That is all what I am asking. In our micromolecule and even our small molecule, which particular feature of our molecule is drug-like and which our feature is not very important for drug action. That is not what I am recognizing. That is what is pharmacophoric feature. Just by looking at the 2D structure of my molecule, I can realize pharmacophoric features. This is what is known as grasping power. Every one of us teachers, as teachers, we have cultivated this habit. The moment we cultivate this habit in our student, we become very good teachers. Okay, so to cultivate this habit in 2 dimensions, we have books available, we have structures available, we have some essays available, we can download the research articles and we can read and tell them. But we need to understand it in 3 dimensions. Then we will be able to very successful. That's what I'm going to show in the coming few slides. The very first thing is, probably I hope you are able to read this. It is what we have done is a rapid racemization in thiazolidinediones, a quantum chemical study. This is what we have done. And I'm going to show you the structure of this. I hope you can see the structure. The colors are probably not very impressive on this screen, but in big screen they are. I hope you can see the structure. Thiazolidinedione ring has got one sulfur, one nitrogen, and 2 carbonyl groups, one asymmetric carbon center with there is a star mark. Using this asymmetric carbon center, this molecule undergoes racemization. What is racemization? Racemization means R isomer is getting converted to the S isomer and S isomer is getting converted to the R isomer. So this message is given to us. Suppose this message is given to us. What to do? How to utilize this information? First thing is that we have to question. What is meant by racemization in 3 dimensions? What is meant by racemization? R isomer. What is the R isomer in 3 dimensions? What is the S isomer in 3 dimensions? Okay, we should look at it. Once we look at it, then why the racemization is happening? You have to ask this question. If racemization is happening, somebody told, yes, it may be happening. R and S conversion is happening. Why it is happening? Is it happening everywhere in our body with every molecule? No. Only in this molecule it is happening. It is happening in ibuprofen also. Like the ibuprofen which we use for our simply for our headache, as an SCID we use. And then that ibuprofen is also undergoing racemization. This is also undergoing. How many total molecules are undergoing racemization? So we have to expand the question. Why it is happening? This is the next question. Can we address it in 2 dimensions? Can we address in 3 dimensions? This is what we have done. We try to look at the molecule in 3 dimensions inside the molecule. Inside the molecule means what? Outside the molecule means what? Let us take the example of our own Professor Anand. If Professor Anand is, you can see him, you can meet him. When you meet him, if he is smiling, you really know that he is in a happy mood. He is going to teach us something very, very important today. Suppose he is in an angry mood. He will be, yes, from the 3D, from his face, you can realize that he is in an angry mood. Today, we are going to have a big lecture on how to behave in the classroom. Not on the chemistry, maybe, probably. Okay, you can easily guess. Similarly, our molecules are smiling. All our molecules are smiling. There is a surface to all our molecules. What is inside? There are bonds. Just like our teacher has got books in his hand, our Dr. Professor Anand has got some models in his hand or some innovative things in his hand is bringing to the classroom. So likewise, our molecules have got bonds. They are important tools to define the bonds, molecule. Now, before that, inside the molecule, there is something. Just like inside our Professor Anand's mind. In his mind, what is inside? Okay, today I am going to teach computer-aided drug design. It is in his mind. He is entering the classroom. You don't know what is in his mind. Okay. If you behave very well, if all the students behave very well, he will teach what he planned to teach. If all the students are telling that सर इलेक्शंस आ रहा है, सर क्रिकेट मैच है आज, सर वर्ल्ड कप चल रहा है, सर हमारा हैदराबाद टीम हार गया, if we start telling like this, he will also say students are not in a good mood, so I will not teach what I want to teach. So what is inside him? Similarly, let us ask the question, what is inside our molecule? Inside our molecule, there are electrons. Inside our molecule, there is energy. Inside our molecule, there are rearrangements. Inside our molecule, there are conformational changes. Inside our molecule, tautomerism. Inside our molecules, electron jumping. Inside our molecule, electron Coulombic interactions. Inside our molecule, we have covalent bonds, coordination bonds. Inside our molecule, we have electron-rich center, electron-poor center. Inside our molecule, we have pharmacophoric features. If we look at all of them in 3 dimensions, we are going to get a lot of advantage. Okay. If we know our teacher is coming happily to the classroom, he is enthusiastically ready to teach today, and if we reciprocate appropriately, the teacher will deliver his lecture very efficiently. Exactly like that. Our drug will deliver what it is intended to deliver. If we realize and if the macromolecule behaves properly with reference to the small molecule. That means the drug, when the drug is interacting with the macromolecule, the drug will do the necessary conformational change on the macromolecule, and then it will be responsible for the release of a secondary messenger or giving a secondary signal transduction or initiate cascade of events and many things. For example, insulin receptor. Insulin receptor, we know insulin binds to insulin receptor, then the message goes into the cell. Then it's telling that insulin is available. If insulin is available, that means GLUT4 gets activated. If GLUT4 gets activated, the GLUT4 goes to travels all the way to the cell wall and captures glucose and brings it into the cell. And when glucose comes into the cell, glucose is divided into CC bonds in glucose gets divided. And when the CC bonds are broken, energy is released. That is how we all get energy. Okay? The food that we are taking is going inside the cell in the form of glucose only. Who is responsible for that? GLUT4. For GLUT4, to activate the GLUT4, what do we need? We need insulin. Where does insulin act? Insulin acts on insulin receptor. So this is a beautiful cascade of events. Very, very beautiful description we can make. Just like in our Bahubali movie, Bahubali can lift Shivling. He can bring the Shivling from simple water somewhere else to the directly below the water. Okay, we can also do, we are nothing less than Bahubali in drug discovery. We can do that if we can think in 3 dimensions. And we think inside the molecule, that is about the drugs. And if we can think on the surface of the molecule, that is about the electrostatic potential. Electrostatic potential, I'm going to show some figures here. So that's what we have done. We tried to find out the energies using quantum chemistry, energy values for tautomerization. We calculated. We found that energy value are 27 kilocalories. That is very large. That means tautomerism cannot happen in this molecule. If a compound has to undergo tautomerism, it should have less than 10 kilocalories energy. Here energy required is 27 kilocalories. It should be about 15 kilocalories. required, but here it is 27. That means tautomerism is not happening in this molecule. It has become very clear from quantum chemistry. What is quantum chemistry? Nothing but solving the Schrodinger equation, that is electronic level information of the drug molecule. That is what was done, and it has been understood. There is lots of meaning in this table. I will not explain all that. Similarly, I can do tautomerism after a reaction called S oxidation. Once S oxidation happens, I will try to do the tautomerism. Then after S oxidation, the tautomerism energy is reduced to 14 kilocalories per mole. That means before before tautomerism, it was before S oxidation, energy is of the order of 26, but it is reduced to 14. 23 became 14, for example. That is telling us that tautomerism is possible. For example, here barrier for a 1,2 hydrogen shift, 1,3 hydrogen shift is 75 kilocalories before S oxidation. It became 38 kilocalories after S oxidation. That means our here this particular figure is showing that the red curve is before the oxidation, the green curve is after double oxidation, but the blue curve is after single oxidation in thiazolidinedione. So that means if our drug molecule is undergoing metabolism with the help of cytochromes, that is also single S oxidation in the rosyglutathione molecule, drug action is happening. Otherwise, tautomerism is not, rapid racemization does not happen. You can give this information, you can publish this paper very well, and this was published in Journal of Physical Chemistry by a PhD student. At that time, she was a PhD student. Today, Ms. Khanna is a very senior, successful scientist working in Bombay in pharmaceutical industry. So why she is able to find this secret? What is that secret? The energy required for the rapid racemization is 80 kilocalories per mole when there is no S oxidation. But when you do single S oxidation, the barrier is reduced, as in the case of blue curve. And when you do double S oxidation, the energy increased again, to as in the green curve. So blue curve, that means single S oxidation, is responsible. Single S oxidation with the help of cytochromes is responsible for the rapid racemization, and thus this is the pathway in which the drug is acting when it is supplied to the human body, and that is how we can explain the science behind the roglutathione, rosyglutathione, pioglitazone, and PPR gamma. This is a very important message. By looking into the molecule, inside the molecule means I already told you electrons are there. Inside the molecule, there is energy. These are the energy curves I'm showing. Red, blue, and green. Beautiful figures will tell you the story of the molecules. Now, so what is the message? The message coming from that is that compound 1S, that is the S isomer of the rosyglutathione, and can undergo rapid racemization only after the S oxidation. Before S oxidation, it does not happen. That means S oxidation is happening where? With the help of cytochromes. Where are the cytochromes available? They are available in the in vivo condition. That means this reaction, that means rapid racemization, can happen only under in vivo condition. It cannot happen in vitro condition. That is a beautiful extrapolation, and that is true experimentally. That is proven. Then this reaction cannot happen in your laboratory because cytochromes are not there. Until and unless you add cytochromes in your reaction mixture, this particular rapid racemization does not happen. How did you know? By looking into the molecule, looking at the energy of the molecule. Now, can it happen in oxazolidinedione? No, it cannot happen because in oxazolidine ion, sulfur is not there. Can it happen in selenazolidine ion? Yes. Selenazolidine, selenium atom and sulfur atom of the thiazolidine ion behave very similar. Thus, this particular rapid racemization can happen in selenazolidine ion, but cannot happen in oxazolidine ion. How we are able to beautifully, one information is giving us lots of information, and that is beauty of the looking inside the molecule, looking at the 3D structure of the molecule. Okay, so that is how I am inviting all of you to the 3D thinking. So this work was proven to be true in 2004, immediately after our original publication. Now I am going to talk about another technology. The first technology I discussed was quantum chemistry. Quantum chemistry can be solved using computer-aided methods. There is a software called Gaussian software. We can buy. It is not very costly. It is only about 1.5 lakhs. You can buy. It is easy to learn. You can come to me if you want to learn how to use it. Okay. So almost everybody can do it at home if you invest only 1.5 lakhs and one laptop. Okay, your research started working. Okay. Another example I'm giving is another computer drug design technology I'm introducing to you now is called QSAR. This you must have heard about it. Most of you must have taught about QSAR. This is very well written in my chapter also. You can read that QSAR is quantitative structure activity relation. Very easy. We know it. What is QS? What is QA? What is QR? It is nothing but a structure should be expressed in quantitative terminology. The activity should be expressed in terms of numbers, and relationship should also be expressed in numbers. Okay. Suppose we have friendship. Friendship between myself and Professor Anand Kumar. The friendship is 20 friendship. Is it correct? Friendship is 20. Can we mention like that? Yes. Sometimes, yes, we can mention. If we are going to the movie together, if both of us like Prabhas, both of us like Virat Kohli, and both of us hate Rohit Sharma, cricketer, suppose, then you can quantify how much is the friendship between these two professors. Okay. So likewise, if you can quantify a relation, if you can quantify the structure of our drug molecule, if you can quantitatively express the activity, that is IC50 value, MIC value, these are quantitative expressions. The structure volume is a expression. The PIC50 value is a quantitative expression. Then you have the length of the molecule is a quantitative expression. Then the surface of the molecule quantitative. The energy of the molecule is quantitative. If I can do that, I can develop QSAR, quantitative structure activity relationship. I can develop with the help of statistics. I told you we use mathematics very easily. We are very clever people. We are learning statistics and we are using QSAR is nothing but statistical approach. Artificial intelligence is also statistical approach. I'm coming to that in a few minutes. Now I'm going to teach you the technology called comparative molecular field analysis. It is called COMFA. COMFA is a very well recognized QSAR technology which is being used by many scientists across industry for drug discovery. People are using that in a very big way. How to use that for PPR gamma and PPR alpha dual activated design. So there are several compounds called dual activators. We want to develop a conform model. For building conform model, we need the structure building methodologies. That is, the 3D structure building methodologies are required. Molecules which are structurally aligned with each other. That is a 3D activity. 3D cubic lattice has to be identified with a grid value of 2 angstrom. That is a 3D activity. Calculation of the integration of the energies has to be performed. Steric based energy as well as electrostatic based energy we need to reduce. Then we have to do many steps, and we have to do statistics called partial least squares approach, and then we can do that. So these are the molecules which are experimentally known to show dual activity. I want to design many more of them. So we have developed a model called alpha model. That is PPR alpha model. Then we developed a model called PPR gamma model. Then both of them were very successful, very good. But the but this that is where the perseverance comes into picture. Perseverance means patience. Patiently pursuing a particular thing is what is called perseverance. Let us take the example. Every research scientist is having this particular quality called perseverance. What is that? Perseverance is the same thing what Bahubali showed in this movie. What did he found? He found a mask. Just behind the mask, he wanted to go behind that. He he imagined that one beautiful lady must be waiting for him above the hill. But he pursued it. He just did not dream. After dreaming, he continued to work on that. Work on that, he was failing many times. But he was doing the same thing again. His mother was telling not to do it, but he was still doing it. Okay? So that is what is perseverance. If you do show perseverance, that's what I am going to describe here. PPR alpha model was very good. PPR gamma model was very good. But the team which is working on this topic, they said that we are not happy. We want a dual model. They have created a model called dual model, and that is called D model, by including simple formula on the rosicular zone. They have used quantum chemistry using PM3. They have used charges. They are known as Mulliken charges. They have done all that. And then they have looked at the 3D structures of the molecule. Look at the 3D structure here. Superimpose many molecules onto one to other, you will know where are the differences. The differences give some information. That is what we are going to utilize. Which particular difference is leading to greater activity, which difference is leading to smaller activity. That is what is nothing but comparative molecular field analysis. That is what is the QSAR. That is what is COMFA. Now, what is this? Just like we keep several chains, one over the other, inside, several vessels, several spoons, several things inside our kitchen, many things we want to make space on the table. We superimpose many objects in our kitchen. Similarly, if we superimpose molecules, we get this kind of a representation, which easily tells us that this is the region where the molecules are similar. This is the region where the molecules are not similar. This is the 3D arrangement, and that is going to help us in coming to details in the next slide. We can do statistics, we can do contour map analysis, we can do molecular docking, we can design new molecules. That is what has been done. This is the statistical result, and this is the colorful result in the form of the 3D structure. Look at the 3D structure of rhodesiglitazone. The sulfur is not visible, nitrogen is not visible, but this molecular structure is visible. And then what is on the surface of that? The surface, I told you a few minutes ago, every drug has got a surface. This is the surface of PPR gum, rosiglitazone. It is telling that this is a region where there is some information. Here, green region, there is some information. Blue region, some information. What is that information? This particular figure is telling that if I take this corner of rosiglitazone and if I modify, I will get improvement in PPR alpha activity as well as PPR gamma activity. If I make improvement here, I will only get PPR alpha activity improvement. If I modulate here, I will get only PPR gamma activity improvement. But again, if I come to this corner, I will see PPR alpha activity as well as PPR gamma activity improvement. Because we can do this kind of improvements, we can do drug discovery. Now, taking rosiglitazone as a lead molecule, can I design new molecules? Yes. Now I know what to do, where I should change, where I should not change. Here, I should not change anything here. If I change anything, it will increase toxicity, it will not increase the therapeutic action. So that is clear. So I should not do anything. Only I should do some change here. I should not do change here. That means I know the secrets. Okay. So this is how we can do drug discovery using computer-aided methods. QSAR is a technology which directly helps in drug discovery, and this is what we are doing for the last 30 years. Your ma'am told that we have published something like 281 papers. This is because we know this secret. That's all. Nothing more. We are not great people. We are just understood the secret in terms of 3D thinking. That's all. Secret. That's all. I'm telling you the secret. Okay. I am telling you the secret. You adopt this computer method on your laptop. You don't have to go too far away. The same laptop which you are using to watch beautiful movies. You will do the research. Okay. Next. I'm going to go to another technology. This is the same technology, how we have succeeded and how we publish results and things like that. I don't want to emphasize on that. I will go to next topic called pharmacophore mapping. Look at my PPR gamma. This is my PPR gamma. PPR gamma, this is a surface. This is a window. From outside the PPR gamma, I can see a window. Through the window, I can see rosiglitazone. How rosiglitazone is sitting pretty inside the PPR gamma. If I take the cross section of my PPR gamma and see the surface, here you can see my rosiglitazone is comfortably sitting inside the PPR gamma. It is in a U shape, three-dimensional shape, but it is sitting inside the cavity like a U shape. It is not straight. PPR rosiglitazone is looking like straight. No, it is not. It is having a 3D structure. It is occupying a U-shaped arrangement inside the cavity of PPR gamma. How do I know? I have to think in 3 dimensions. That's all. This is the secret. Simple secret, nothing more. Very simple. Any scientist who can think in 3 dimensions will be successful. Any scientist who think in 1 dimension or 2 dimensions is going to be a failure, guaranteed. If we are sending Chandrayaan today, if I can imagine where it is going to land, what is the surface? What is the 3D structure of the surface? Is there water? Is there a hill in region? Shall I land my Chandrayaan on a hill? Or should I land it on a tree? Is there any tree there? Okay, if I start asking this kind of questions, then I will know what to do. If I can think 3D, as ISRO scientists can easily make a plan. Similarly, you as a drug discovery scientist, you should be able to think in 3 dimensions. That is what this figure is showing. 3D structure along with the cavity inside the gufa, what we say in Telugu, guha, guhalopala, the rosiglitazone zone is sitting. Similarly, here this surface is showing that this red, blue, yellow colored surface is a cave. Inside the cave, our rosiglitazone zone is entering and comfortably sitting. I am just showing the cross section here in this figure, showing that the pink colored rosiglitazone zone is comfortably sitting in a U shape inside the cavity of the PPR gamma because I know it because I am showing you in 3D. So using this, I can create pharmacophore maps. I can use this for virtual screening. I can create, I can start with a data bank of 3.5 lakh molecules. And here, the 3 different databases, almost 5 lakh molecules. And then we go to pharmacophore 1. Then we can screen to 1700. Further, I can screen to 1092. Then further screening using Lipinski rule of 5 makes it 533. Further screening gives rise to total only 19 molecules. From 5 lakh molecules, I come to 19 molecules within 1 week. I'm not doing experiments here. I'm only doing for computer-aided drug design to eliminate many useless compounds and identify useful compounds. 19 compounds. I ask my organic chemistry friend, can you please synthesize these 19 compounds? He will say, yes, I will do it. You ask him to synthesize 5 lakh molecules. He will say, wow, bhago yahan se. I will not do it. Get lost. He will not even respect us. If you ask him to synthesize these 19 compounds, he will say that yes, we will do it. And then we'll do drug discovery. And then these are all many more data. And these are all the varieties of molecules. This is the U-shaped drug inside the U-shaped cavity. This imagine it is in 3 dimensions, and then you will understand this better. It is a mu-shaped cavity is present inside PPR gamma, and my drug molecule rosiglitazone is occupying in a U-shaped manner because I know it because we are able to think in 3 dimensions using the computer-aided drug design methodologies. This is called docking result. The same rosiglitazone, when it is docked inside the cavity of the macromolecule, it is how it will look like. Now, what is docking? Docking is a computer-aided drug design technology which can be done on a macromolecule using a small molecule. And when we successfully do, we know how the drug is sitting inside the macromolecule. Very cleverly, you can do that. This is very similar. One more time, let us take the example of Chandrayaan. How the Chandrayaan is right now, how far it is from the moon. After 6 days, if it is going to land, how it is going to go close to the surface of the moon. What is the speed with which it will go? Will it actually land or will it bump into moon? Will it fall? Will it fall down or will it land? These differences you can know by doing by doing the modeling. They have done it already. They have done modeling first and then only they sent the Chandrayaan. Now we can do the modeling using the 3D docking approaches, knowing these kind of deterrents. Where is the hydrogen bond? Where is the hydrophobic interaction? Where is the van der Waals interaction? Where is the Coulomb interaction? Where is the hydrophobic interaction? Where is the covalent bond? Where is the coordination bond? Which particular interaction is helping the interaction between rosiglitazone and the PPR gamma? Can I design a molecule which will be behaving very similar to that? If I can design a molecule which interacts with PPR gamma in the same way, my work is done. I have got a new drug molecule which we can design using the computer-aided methods by thinking in 3 dimensions. This is the docking pose. Now I will go to another docking pose. These are the class of molecules. These are another class of molecules. Again dock them, and then finally you perform the drug discovery research. And I am sure this is what we have succeeded. I am not showing all the results because it is a trade secret. Which molecules have succeeded, which molecules have failed. I will not tell those. But I can think now again in a different way. Now, how can I use all this? Till now, we discussed quantum chemistry, CADD method. We have discussed molecular docking, CADD method. We have discussed QSAR, that is also computer-aided drug discovery method. We have discussed surface properties, CADD. We have discussed virtual screening, CADD. There are so many technologies under computer-aided drug design. Now what I will do, I will stop this lecture and will start another lecture. That is on artificial intelligence, because that is a growing topic. So some of you may ask, can we use artificial intelligence for drug discovery? Yes, that is my answer. I am opening another lecture. I hope you can see it. Okay. Can you see my new lecture?
No, sir.
Should I share again?
Yes, sir. Visible, sir.
Okay, sir.
Yes, sir. Visible, sir. Slide is visible.
The artificial intelligence. Almost everybody in this world is talking about artificial intelligence. Every day you open the newspaper, it is artificial intelligence. Everybody is telling that, okay, we will have robots, we will have our Rajnikant, we will ask Rajnikant, he will tell that using artificial intelligence, he created Chitti, and Chitti, he became two parts, Chitti 1 and Chitti 2, and then we have seen many things that Chitti can do. Similarly, artificial intelligence can do many things. In drug discovery, how to use it. For this purpose, I am going to give brief introduction. That is, artificial intelligence can be used for drug discovery, drug design, drug disposition, drug toxicity, drug repurposing, drug metabolism, and many more things. Clinical studies can be done using artificial intelligence. Pharmaceutical management can be done. Natural product chemistry can be done. Many things, many things can be done. Ayurveda can be done using artificial intelligence. We are doing it already in Naipur Mohali. So for all this, what do we need? The basic thing is think in 3 dimensions. First thing. Okay. So some people are, it's not very easy to tell them. Sometimes we teach in a classroom, very young boy of 20 years, if he comes to us for some suggestions. We tell him, "Arey boy, you build your confidence. You will be able to win the world." Then this boy, 20-year-old boy, thinks, what is confidence? We get confidence. Teacher told very nicely, you build your confidence. How to build confidence? This is questions that are in the minds of our young students. Similarly, if I am telling you to think in 3 dimensions, but how to think in 3 dimensions, you are getting the question. For that only, I am telling your laptop is very useful. You can download software to help cultivate the habit of thinking in 3 dimensions. Anyway, here in this lecture, I am going to tell you how artificial intelligence is being utilized in our institute for in drug metabolism and in drug toxicity we are using, and that's what I'm going to show you the example. Let us first see what is artificial intelligence. It is a science and engineering of making intelligent machines, especially intelligent computer programs. It is nothing but a computer program. Let us don't think it is a great thing. Artificial intelligence, people talk about it as a very great thing. Nothing great about it. Suppose you don't know what is a, oh, if you don't know what is a mobile, for example. Can I imagine that you don't know what is mobile? I should not imagine. This is absolutely wrong. But suppose instead of that, I imagine something completely different. That rubber stamp. This is in my hand. Okay. So there is what is in my mind, what is in this word, rubber stamp. There are different things. If I say pin, what is a pin? I say word pin. Pin drop silence wala pin. What is the pin you understand? You understand a pin differently. And I am showing you a pin which is different. This kind of pin. So there are some kind of differences in our own communication. So if we can communicate properly with the computer by removing this kind of differences, computer will become intelligent. That is why we need to understand what is the syntax, what are semantics. Semantics is about the grammar of computer program, and syntax is the meaning of a computer program. Okay, every line of computer program has got a meaning. That is what is known as semantics. What is the grammar? It is syntax. I think I told wrongly earlier. Semantics means the meaning. Syntax means the grammar. Okay, if we can understand that, if we can play with it properly, we will get artificial intelligence. This was done in 1960s. When the Fortran language was invented, they invented another language called Prolog. It is not sufficiently popular. But later, they invented a language called Lisp. It's called list processing, another language. Likewise, they have developed many languages. Out of that, one of them is known as artificial neural networks. It is not called a language, it is called a technology. Then that is further developed somewhere in 1980. I hope we are back, and hope you can see me. And
Yes, sir.
now,
Proceed, sir.
under under the artificial intelligence, there are several things called machine learning methods, knowledge-based methods, artificial neural network methods, deep neural network methods, convolutional neural network methods, support vector machines, like that. There are so many subtopics and technologies. All of them are being used successfully for drug discovery. Artificial intelligence depends on mathematical logic. So, mathematical logic. Prolog and Lisp are the programs which work on logic. And drug discovery can be shown like this. A big circle is artificial intelligence. Under that, there is a subtopic called machine learning. Machine learning overlaps between KBS, genetic algorithms, deep learning, and fuzzy systems. Under the machine learning approaches, there is SVM, ANN, DNN. These are all subsets.
Stop.
Okay, so hello.
Start చెప్పండి సార్.
Oh,
సార్ ఏ సార్ ఏమైనా మాట్లాడారు కదా వీళ్ళు
Sir, please continue, sir.
Okay. Fine. So these are the three well-known knowledge-based systems. What is knowledge-based system? Google is a knowledge-based system. We are using every day Google. So this is one of the artificial intelligence. So we are using every day artificial intelligence. Don't think that we are far from it. Okay, when we are your mobile, when it is automatically showing, you want today we have seen Chandrayaan Wala on news item. Tomorrow also it will show Chandrayaan news item. Why? Because it is using artificial. So it realizes that you like to read about Chandrayaan, so it is showing another news article on Chandrayaan immediately, quickly. Every time you open, it is showing that this is nothing but artificial intelligence. So likewise, that is actually under within the artificial intelligence. What is that subtopic? That is what is a knowledge-based system. It is gathering the knowledge, what you like. So accordingly, it is showing that. Okay. So similarly, if there are several farm GKB, daily med, super target, these kind of things are already available in artificial intelligence knowledge base related to the drug pharmaceutical sciences. So I am not going to explain what are these, but I will go to the next topic, that is the genetic algorithms. Genetic algorithms behaves like genes. From one generation to another generation, when we carry the knowledge, that knowledge is what we call as genetic algorithms. If we can write a program to mimic how the knowledge is carrying from one generation to another generation, that is nothing but genetic algorithms. Suppose I start with 20,000 molecules. I will remove some of them and I will add a few and I will get a better generation. Then I will again get a better generation. Next generation is better than the old one. Likewise, I will do many generations. In the first generation, there are 20,000 molecules for PPR gamma activity. In the last generation, there are only 20 compounds which are which are important for PPR gamma activity. These last 20 compounds are really useful out of the first 20,000. This is nothing but virtual screening through genetic algorithms. Every generation is better than the previous generation. This is the idea. So that is the logic behind genetic algorithms. Then we can go to many more things. Without explaining all this, I will go to the next technology. What is that? Next technology is the machine learning methods called support vector machine. Support vectors are some kind of numerical parameters which are related to each other. Related parameters. Suppose if I take idli today, I will also probably have taken sambar. But if I taken paratha today, probably I have taken achar with it, maybe. So like that, there are related knowledges. This related information is known as support vectors. These vectors are utilized in this kind of graphical manner, and we can classify the existing data, and we can employ that technology in drug discovery. So this is this figure is showing artificial neural networks. This is called a neuron. Where is it? I should take a neuron. Here. This is a neuron. This is another neuron. This is called hidden layer. Neurons in the hidden layer. This is called the neurons in the input layer. This is called the neurons in the output layer. So in this process, what happens is that some numbers are given to each of these neurons, and these numbers are adjusted through training. We can train. Suppose I can train every human being. I can tell that this is, suppose I tell you, this is a red pen. This color of this pen is red. Do you agree? You may not agree. because you are already trained that this is not red, this is kind of a gray. But suppose I tell a small boy and I bombard him with the knowledge, this is red. Okay? Suppose I give the wrong information. So the boy gets trained like that only. Similarly, I can train a neural network. So if I can train a neural network, I can use it forever in the next few years. That is the purpose behind artificial neural networks. This is a network called neural network because each of these box is a neuron. This works very similar to the way our brain works. Next, I go to another topic, deep neural network. Deep neural network is a part and parcel of machine learning methods, and it is a part and parcel of this kind of a network. Several neurons are networked. Here, this is one hidden layer, another hidden layer, another hidden layer, several hidden layers. Each of them, for example, this may be 1024 neurons here. This may be another 1024. This may be 128. And the information is coming from one layer to another layer, to another layer. These are all numbers, back propagation, feed forward. Somehow we will be able to train this network, and this network will become useful for us. This is mm, this kind of thing is called deep neural network. We can use it in drug discovery. Similarly, drug metabolism is being carried out with the help of artificial neural network, and this kind of, for example, here for here, CYP2C9, CYP2D6, CYP3A4. These are all the enzymes which are drug metabolizing enzymes. Which particular enzyme is more suitable for my drug? I want to know. I have identified a new drug. Which enzyme is metabolizing my drug? If I want to know whether CYP2C9 or 2D6 or 3A4, to do that, I can perform I can perform the mm, I can perform the artificial intelligence tool development. That is what has been done, and I'm showing. So this is what was done somewhere in the current drug metabolism. It is published, and Genosite, Metapred, ID site, Smart CYP, FAME, Star Drop. These are all the tools. They are not very. Some of them are free. Some of them are costly. You may have to buy, but they are all metabolism studies. So this is SVM, support vector-based machine-based approach. This is artificial neural networks-based approach. Both these methods are giving the same answer, that this particular site is easy to metabolize, and these two sites are easy to metabolize, whereas this site is not so very easy to metabolize the intelligence. Similarly, lots of artificial intelligence has been done. I don't have time to discuss all that. Sorry for the disturbance during the presentation, but this is a problem we have to face in India. So artificial intelligence requires data collection and digitization. This is one of the first steps. Next, organization of the data and available information. Second step. Next is the knowledge synthesis. Knowledge synthesis means generate the knowledge from the existing data. Data is abstract. Data does not tell you what is the information, but we have to extract the information. That is the process of artificial intelligence. And then once we get a meaningful output, we can say that we are succeeded. Many people are using, you can search in the internet, generative artificial intelligence in drug discovery. If you type this in Google, what is it? Generative artificial intelligence in drug discovery. If you type these words, you will get many research articles, and I am sure you will be able to learn a lot. So, but I have to stop somewhere, and I want to conclude that artificial intelligence and machine learning are being used extensively in academia for design, disposition, delivery, and the discovery applications of the drugs. Artificial intelligence is also being used for quality control. Artificial intelligence is also being used for quality by design. Artificial intelligence is also being used for industrial processes. So I wish you will be able to teach and learn this, and I'm sure the entire society will get benefit from this kind of a knowledge. Thank you very much.